My end, please somebody confirm that we're live. As soon as it's preparing, setting up. My end, please somebody confirm that we're live. We're okay, yes.
Yeah, it's live, yes.
It's live, it is. So yes, welcome you guys. We'll give it another minute. If you hear lots of banging, they're replacing the roof at home. So as soon as I finish briefly introducing our host, who will introduce our speaker, I'll mute my microphone.
Okay.
I was telling Lina, it just blows my mind that they replaced the roof in one day. So yeah, I'll let you know how it goes.
Eduardo, if you
Well,
lived
it's better to be better rather than be without a roof for a few days. So yes.
Sorry, Tony.
Eduardo, if you lived in Texas, you wouldn't be surprised that they do that because we get these freak hailstorms like literally 2-3 times a year with hail that's this big, and there are companies that are just dedicated to replacing people's roofs after hail.
Right.
Crazy.
Okay, I think being 11:01, we can probably get started. Welcome you all. Welcome our YouTube community. I take the opportunity to invite you to join us via Zoom if you so like. There are no passwords. You can just jump in and attend live, and we get to see you and you get to see us. But we also like to have the YouTube community. So please, also you can post questions if you're watching this. So today's meeting is somewhat special because we have one of our former grad students responsible for holding the speaker. And the reason I say that she was a grad student is because the person who's going to be hosting the talk today is our Dr. Lindelani Makuya, who got her PhD from the University of Witwatersrand in Johannesburg, and she's been working with Karsten Schraring for many years as she was doing her research. So the plan is that I must stop talking right now and defer to Linde, who will take it from here in introducing our speaker and doing what we normally do on every fine seminar. So Linde, once again, congratulations on this huge accomplishment, and please take it from here.
Yes, thank you, Eduardo. I was not expecting the picture. But luckily it was a nice picture.
Blame me,
So
it wasn't my idea. Somebody told me, you can guess who. Somebody told me, just show the picture. I'm not gonna say who, you can guess.
Yeah. Okay, so good day everyone, and welcome to today's Fine. So this is the last one for this season, and I'd just like to first of all thank all our speakers and all our participants and guests over the season. I believe it has been a lovely season, and if you have missed any of the previous Fine talks, you can always watch them on our YouTube channel. All of them are always there, and you can catch up at your own time and own pace, or even use them as some people are doing as a teaching tool, because I think it's very good talks that we had this season. And the Fine Spring for 2025 will begin at the end of February, and I was told that the list is already filling up. So we'll release the program for that towards at the beginning of 2025. So we're still looking forward to having you join us still for another season of Fine. And today we have Manjari Jain, who is an associate professor from the Indian Institute of Science, Education and Research in the Department of Biological Sciences. And her research mainly focuses on working in the tropics, and she works on both insect and bird model species, and she does this to understand signal evolution. So I won't talk much about it, her work. I'll let her do all the talking. So over to you, Manjari.
Thank you very much, Linde and Eduardo, and all involved in the fine seminar series. Indeed, it's a fine series of very interesting seminars, which I have been using for my teaching. Like Linde said, I do work on both insects and birds. Can you just confirm if you can see my slides? Okay. And what I want to do today, I know there are many who have talked about bird model systems in the seminar series, and I thought I will talk about insects who are not social, but they do aggregate in large groups to communicate with each other. So this is one topic which is very close to my heart, the cocktail party problem, as to how animals communicate using sound and how they face and solve the challenges of communicating in large, noisy choruses. So just to orient you a bit, I work at the Indian Institute of Science, Education and Research at Mohali. The institute is situated in the foothills of Western Himalayas, and we are not too far from Delhi. I also want to take this opportunity to advertise the behavior meeting, which will be held in Kolkata in India next year. And I hope all of you will be joining. I hope to see some of you there. So my larger interest has been to understand various factors that drive signal evolution. And for that, I am working on social babblers to understand social behavior, of course, but also how they communicate, how their communication impacts their social interactions or mediates their social interactions, but also how sociality may have influenced the kinds of calls they produce, sound complexity in the context of understanding evolution of language. But I will not talk about that today, and instead I will focus on crickets and katydids and talk about the work I have been doing on crickets and katydids to understand various factors driving signal evolution. So I have looked at how habitat structure may drive acoustic signal structure from the point of view of the acoustic adaptation hypothesis. I have also in the past examined and continue to examine how biological noise may be shaping signal structure, or how signal structures may have evolved to escape masking, or at least reduce masking avoidance. This is what I will talk about today. I have also worked on and continue to work on mate choice and sexual selection and understanding signal evolution from that point of view, and also natural selection from the perspective of predation and parasitism, as well as now currently I'm also working on how infection may affect signaling behavior. But these are all, while they all seem like many different questions, the theme is a singular thing that we are pursuing. We're trying to understand what are the various factors that shape acoustic signal structures. Acoustic signal structures in crickets that have very different kind of calls, and acoustic signal structure in birds which also have very different kind and context of communication. So since today I'll be talking about crickets, just to familiarize you with what crickets are. Crickets are basically nocturnal ensiferan insects in which males produce sound by stridulation, by rubbing their wings together. And If you look at the four wings of males, you can see very distinct sound-producing structures. There is the harp-shaped structure on the wing, which is well called as the harp. Then there is a vein which carries a row of teeth underneath, which is known as the file. Then there is a hardened edge of the wing, which is referred to as the plectrum. And there are other areas of the wing which help the animal amplify certain resonant frequencies. But these structures are only borne by the male wing. These are absent in the female wings. So the females are silent, but of course they exercise mate choice. So you will have male crickets singing and females approaching the males just by sound alone. And if you look at the sound structure, crickets have chirp-like calls, but they can also have trill-like calls. A bunch of syllables put together is known as chirp. And we measure various parameters of the calls of the cricket, such as chirp duration, chirp period, syllable duration, syllable period. These are all temporal parameters which define how energy is distributed over time in the song. Spectral parameters, on the other hand, tell us how energy is distributed over frequencies. So, understanding whether all the energy is concentrated in a narrow range of frequencies, the narrow band call, or if energy is distributed across a very large range of frequencies, where then it would be a broadband call. So, true crickets or field crickets are typically very tonal singers, and they have most of their energy concentrated in a very small range of frequencies. But katydids, that are, well, you can say cousins of crickets, also ensiferan insects, they typically have very broad band calls. So this is what a typical field cricket sounds like. So the problem that I have been interested in is to understand how crickets and, well, other animals as well deal with the problem of what is called as acoustic masking, which is simply when sounds overlap with each other both in time and in frequency. So as you can imagine, this can be from the ambient noise around us, but biotic acoustic masking or by other animals can be either conspecific acoustic masking, where other singing individuals of the same species are masking each other because they are signaling concurrently, or it could be heterospecific masking, where you have different species calling together and they mask each other. Now, this, of course, is expected to impair signal detection by the receiver, localization and perception of variation between the signals. And this also is very commonly known as the cocktail party problem, because it is simply a problem of sound source separation. So if there's a source environment where there are multiple sources of sound, then the target sound or the sound of interest, where is it coming from, to be able to detect it, to be able to localize it, and to be able to decode it, to have all the information intact, that is the cocktail party problem. And this, of course, is borrowed from the cocktail party problems that we face in humans. But as we know, communication by sound is not unique to humans, and nor are the problems associated with it. So the first part of the work that I will present was carried out in the Western Ghats in southern India. The red dot that you can see is a stretch of it's Kudremukh National Park, a stretch of virgin rainforests, which where we studied the nocturnal crickets and katydids for a very long time in the lab of Dr. Rohini Balakrishnan. This is where I was doing my PhD. So that's when I started getting interested in this problem. And we know the forest structure very well because we measured it, and we know that the forests are very heterogeneous in structure. So there is this canopy, then there's a layer of understory, then there's a gap between the understory and the canopy where sound can transmit much better. So for a large part of my PhD work, what I did was to understand how signals transmit in the habitat. But once I was done with my PhD, what I got interested in was to understand that how are animals actually communicating in noisy acoustic environments, even in these tropical rainforests, which may appear like silent places, but they are not really silent because there are multiple species calling together. And we know that there are at least 20 different kinds of species that call together at night. So imagine one species of cricket is calling, and there's another that is calling nearby, and another, and another. What it will result in is these sounds overlapping with each other, resulting in acoustic masking. So these are some of the very beautiful crickets and katydids found in Kudremukh. And we know that they were present in the same season or so. And if I'll just play back the calls of how some of these crickets, not all, but some of them sort of together, and I have jacked up the volume a bit. But yeah, so just warning, if you have very loud volume setting, please reduce it. So this is what a night in the forest would sound like. The question really is that we already knew from some other people's work that Kudremukh has a high diversity of crickets and katydids. And they are all acoustically active. The question was, how are they communicating? We know that, of course, they are communicating effectively, they are all there. The question is to understand the mechanism by how they may be escaping masking, or to at least understand the degree of acoustic masking that they face. So this is what we set out to examine. Now, for any classical cocktail party problem, there are several potential solutions that are possible. The first and the simplest one is temporal partitioning. Different animals may partition in time, so their activity is higher in a particular season or at a particular time of the day, and so they are partitioning in time and they escape, reduce acoustic masking. Or their calling activity can be partitioned, which is in a particular sound window. You may have some species that are calling, but others don't call, and this can happen actively or just by chance alone. Last but not the least is the signal structure itself is such that there are gaps between the calls, and the animals, they may be actually calling in a manner that their signals actually fall largely in the gaps, in the silent intervals. Now, this fine temporal avoidance can happen if they are behaviorally avoiding to call together, or it could simply be the call structure has evolved in a manner that it is a high duty cycle or a low duty cycle call, which means the calls are either very spaced apart, so there's enough silent interval. And so if one such species is calling, another species, if it calls, which also has a low duty cycle call, it may just happen that when they call together without any active avoidance, if you just run the calls against each other, the probability of masking is anyway very low, which is because the kind of signal, the temporal structure of the signal itself. So this is one possible solution. The other, of course, is spectral partitioning. Just like we tune into the radio, the receivers may be tuning into particular frequency bands, and the signalers, the callers, may be calling in particular frequencies. So this is another way of, and a very powerful way of avoiding masking interference. Of course, spacing. So you can space away from the noisy uh, uh, um, neighbor or the source of noise. Edurado can't do that. I think they're right above him, so you can't run away. But animals can. They can space apart both vertically or horizontally in space. And if nothing else works, animals can be loud. So they can actively increase their call intensity, or maybe that they have evolved to have louder calls. So these are the possible solutions to the cocktail party problem. So we wanted to know what is really going on with the crickets in Kudremukh. The first question, of course, that we asked was, do crickets of Kudremukh avoid calling in the same season or time of the day? To address seasonal partitioning and dial partitioning, we know that that is not the case. So most of the species that we studied actually call in the same time of the year and also the same time of the night. So the first two axes of separation is ruled out. So then we asked, what about other forms of temporal masking avoidance, which is to examine if two species A and B, when they call, are they avoiding to call, or do they, the probability of finding A calling when B is also calling, even though you're looking at the same season at the same time of the night, you take a 5-minute time window. In that 5 minutes time window, will you find both A and B calling together? So that we call as the gross temporal overlap. So at a very crude level, like at a very broad level, are they calling together? They are calling at the same time of the night. So between 6 to 12, 6 p.m. to midnight, they are all calling, but within a short window. So masking will only happen when it is sort of happening together, instantaneous. So is that happening? That's what we set out to assess. And for this, we sampled in the rainforest and recorded calls to, and then examined pairwise masking probabilities. So what is the probability that A was calling when B was also calling, and calculated the total incidences of this to get at the probability of gross temporal overlap. Next, what we did was, even if they are calling together, their signal structure may be such that when two calls come together instantly, much of the calls of species A may not be overlapped by species B simply because of the call structure of species A as well as of species B. So for this, what we did was to ask if crickets of Kudremukh avoid calling together at the fine temporal scale. So if there is active avoidance, and we didn't really find evidence for active avoidance, so we started to examine if the signals are such that if you run one signal against the other in time, what proportion of their chirps are going to be overlapped with each other. And then you add time delays to the signals to simulate species A calling first, species B calling later, so on and so forth. And you calculate average fine temporal overlap. So this tells you what is the probability of these two signals overlapping each other if the crickets were calling at the same time window but not actively avoiding each other. So just by chance alone. So therefore, just by the signal structure itself, what is the chance of fine temporal overlap? Now, actual temporal overlap or effective temporal overlap will only be when they are calling, when two species are calling at the same time window. So this is GTO or the gross temporal overlap, and their signals are overlapping because of the structure, temporal structure of the signal. So you multiply both these probabilities to get an effective temporal overlap. We also examined, since we had the calls of all the target species, we examined what proportion of their spectral space is overlapped with each other. So you basically see the spectral profile of species A and species B, and you calculate the area under the curve which is overlapping. So you have the power spectrum of species A, power spectrum of species B, and you calculate how much of this actually overlaps. So that will give you spectral overlap. So we have gross temporal overlap, fine temporal overlap, spectral overlap. So this is what we did. We basically went to the forest. This is my colleague Swati. And we were recording the calls with uh, a bat detector, because we had several crickets and katydids that produce high frequency call. And we were also sampling by ear psychoacoustically. And then we asked, if red one be species A and the green one be species B, what proportion of the calls of species A is overlapped by species B. And as you can imagine, this matrix is not symmetrical. The proportion of the calls of species A that is masked by species B is not going to be the same for most species pairs. So now we have 14 species, and so we go ahead and look at all pairwise combinations. So 182 species pair combinations we had, and these are what the results look like. So at the gross temporal overlap for in a 5-minute window, what is the probability of any two species calling together? This is what the results look like, the first graph. On the y-axis, we have the probability of masking. On the x-axis, we have probability of masking. And on the y-axis, we have the number of species, the frequency, how many species pair had that probability of masking. This is simply that. So as you can see, the first graph is more or less bimodal. So there are a bunch of species pair where the probability of them calling together in a 5 minutes window is anyway quite low. So it's less than 50%. And another bunch of species where this probability is actually quite high. So that is the GTO, and the median for this was about 0.31. Okay. We then examined for these 182 species pair, what is the fine temporal overlap, which is to be attributed not to their calling activity, but to the signal structure. And what do we find? Again, we find that yes, there are large number of species that have very little fine temporal overlap. As you can see, 0.1 has the highest number of species pair having 10% overlap in their call structure. But if you see on the right hand side of the second graph, the FTO graph, you can see there are large number of species who actually have more than 50% and some that are as high as 90% of overlap at the fine temporal scale. But when you multiply both this probability, that is when you get the effective temporal overlap. And as you can see, the third graph, graph C, you can see on the right hand side, those bars have disappeared simply because the probability that they will call together at the same time and they have signal structures which are conducive to masking each other comes down when you multiply the probabilities. So as you can see, not surprising, the values are much lower. What about spectral overlap? Even if two animals are calling together and at the same time, truly acoustically they will mask each other if they are in the same frequency band. So this was a crucial measure for us to make, and we find that there are large number of species that have nearly zero spectral overlap, but there are sufficiently large number of pairs where spectral overlap was indeed non-negligible. Okay, so now we have the effective temporal overlap, which is represented in graph C, and we have the spectral overlap. What do we do next? So we ask that if there is if species that call together at the same time have signal structures which are likely to not mask each other. Okay, so whether there is any correlation between GTO and FTO, we found a significant negative correlation between GTO and FTO, but not between GTO and spectral overlap. So basically, this means that species that had high temporal overlap of signal structures were likely to avoid calling together, but species with high spectral overlap are no more likely to avoid calling each other in the temporal domain. So what does this tell us? Are they avoiding each other or not avoiding each other? Not really. Okay. Then what we found was that if you look at the gross temporal overlap distribution was quite broad, but FTO, ETO, and SO was towards the left, which basically indicates that masking probabilities for most species is anyway quite low, 10% to 30% at max. Okay. Now this I'm talking at the community level. I'm talking about 14 species that are all calling in these choruses, and we are asking, and when we look at pairwise overlap, what is the masking like? So one species with another species. But remember, this is a chorus where there are multiple species calling together, and we are only looking at pairwise masking probability. So in a way, we are artificially already reducing the masking problem for the species, because we're only looking at A being masked by B. But when A is signaling B, C, D, E, F, there are others who are calling as well. So we're not looking at a multi-species level. We are only looking at pairwise interaction. So that was definitely something that we had to worry about. Okay, I'll come to that in a moment. So what we realized was we have accounted for temporal overlap, we accounted for spectral overlap, but the animals are not calling in an anechoic chamber. They're calling in the natural habitat, which has leaves, which has stems, they had boulders, and when the sound transmits to the forest, it will degrade. Our simulations don't account for, our calculations don't account for the kind of degradation of signals that may occur when they are calling. So sound transmission itself is a powerful way of reducing overlap with each other. Okay, so we accounted for that. We took a subset of 5 species which are most likely to experience masking, uh, and we took the most difficult cases, so the absolute worst possible maskers who call together at the same time of the night and they have overlap in their temporal activity. Their call structure is somewhat overlapping and they have high spectral overlap probability, so on and so forth. And then we went and I mapped 10 natural multi-species choruses to assess natural spacing. Sorry, how far the individuals are spaced apart from each other, because in the first two we are not accounting for spacing, we are not accounting for signal degradation, and we measured how loud each of these species are calling to assess, and then carried out transmission experiments in the rainforest to see how far, when one cricket species A is calling, how far will the call transmit anyway to begin with? Maybe it will not transmit so far. So the acoustic sphere, the volume of acoustic space over which the sound can be even heard, is so small that masking is not even real. So this was something that we had not looked at then, but we measured that then. We also, we took tuning of receivers using a sender receiver match model. We did not conduct tuning experiments. And we took these hearing thresholds from the literature and developed a 3D active space model to compute the active space overlap. So you can think of a cricket as a source of sound and a sphere of sound around it because the sound is radiating. And if there's another neighboring cricket, it also has its own sphere. The radius of the sphere is determined by how far the sound actually transmits, which we measured by transmission experiments. And so the radius of each of the species is not going to be the same. Some crickets have sounds that transmit really far. Some others have shorter radius over which the sound transmits. And then we examine how much of the overlap is there in these two spheres. Now, I will spare you the horror of explaining how we did this whole thing, simply because two-sphere intersection would be when you are looking at pairwise interactions. We were interested in multi-species interactions. So you are now looking at potentially five spheres masking each other. Okay, so you can have one focal animal and you can have multiple maskers. Okay, so now we are, and how many maskers are going to be there, that depends upon the natural chorus itself. In the natural chorus, if species A had 5 other individuals around it, then we use that information. So basically we recreated the forest in a simulation, something like this. So you have species A calling, and there are other individuals that are calling nearby, some who may have very small, short distances over which their sounds travel, so they don't even mask the species, focal species. Others who have large enough spheres and who are close enough to the focal individual to be potentially masking it. So we then multiplied the effective temporal overlap with the active space overlap to get at the final value, which is known as the effective acoustic overlap. So this was done for species pairs. So we looked at pairwise interaction, but also did it for multi-species choruses for a given focal species. So each of the 5 species we used as a focal species and examined both pairwise interaction as well as multi-species interactions. Okay, so the EAO accounts for gross temporal overlap, fine temporal overlap, natural spacing of animals, because you must remember spacing is also a powerful way of reducing masking, potentially a powerful way. Sound transmission differences, differences in the calling intensity and receiver tuning. Okay. We had a very interesting time conducting these transmission experiments from the forest floor to the canopy, where the forest doesn't have canopy walkways or cranes, and we did not want to build these walkways or bring in a crane and destroy the forest. So what we did was we established a monkey line in the canopy of the rainforest. We had some people from professional climbers who helped us conduct these experiments, and we uh, examined how far do these sounds transmit in the forest, in different parts of the forest. Okay, so these were the heroes of the story, the 5 cricket and katydid species. This is Mycopoda, this is Pyrmeda, this is Londreva, Phaloria, and this species is not yet described. This we call as whiner because the call is like a whine. As you can see, they have a variety of call types. Mycopoda has calls which has a complex verse-like structure. There's a chirp and then there are 2 clicks. Then Pyrmeda has chirps which have 3 syllable chirps, but you can see it's a low duty cycle call as compared to Mycopoda. Londreva has only pulses or syllables which are repeated. So although it is high duty cycle, you can see it has only a single pulse call. Phaloria has pulses which are bunched together in chirp-like structures, but they are not quite chirp. These are like long trills. And then you have whiners. So as you can see, a variety of call types in the temporal domain, and in the spectral domain, you have 2 really broadband callers, and you have 3 callers that are narrowband. But as you can see from the spectral profile, their spectral profiles overlap quite a bit, and they all are understory species, and they all call at the same time of the night, so on and so forth. So these were the worst maskers in the community. So we picked these 5 worst maskers, and you can see the Mycopoda goes up to 80 kilohertz. So, it's really going into the ultrasound, so on and so forth, as does Pyrmeda. But others are relatively lower frequency callers. Okay, so in pairwise interaction, when we using our simulations, when we estimated what is the probability of species A overlapping with species B. So on the x-axis are the species pairs. You will see that most of the species pair are at the floor. So basically, most of them experience nearly zero masking from each other. When you account for how the signal transmits, how far they are from each other, their calling intensity, so on and so forth. We have not even accounted for, we have not even put into the model that the animals may actually be behaviorally actively avoiding each other without active avoidance, just by the signal structure and the transmission of the sounds in the forest. What we find is that it is nearly a silence in the cacophony. But of course, there are some species pairs that they mask each other very much. But again, like I pointed out, so median effective acoustic overlap in pairwise interaction is close to 0 for 17 out of 20 species pair. But this is pairwise interaction. In the natural choruses, it's not just one neighbor that you have to worry about, there are multiple neighbors. So this is what we then did. We looked at multiple masking sources, and each of the plot is for one particular species. Okay, for species A, species B, C, D, E. And as you can see, it's only for some species where the probability of masking from multiple maskers is rather high. But in most cases, the probability of masking is skewed to the left, it's close to 0. So what we are trying to tell you is that it is almost silence in a cacophony. Now, what we did was, so this was the first part of the work, and we published it in Evolutionary Ecology. For me, this was completely unexpected. I did not expect that the masking levels at the chorus level, where multiple species are calling at the same time, and they are calling quite loud, and they are all existing in the same, space in the forest, masking levels are this low. So then we wanted to know that, what would happen, what would be the effect of color density? How much can this rainforest actually hold? Currently, if there's one focal individual and say there are 5 maskers of belonging to 3 different species, what if you pack the rainforest with more collars? How much can the forest hold? So, what is the effect of collar densities? But also, more importantly, chorus composition. Does the identity of the species in the chorus matter? So, this is the next question we asked. I will not go into too much detail, but this was basically like a cocktail party nightmare scenario. We know that call structure, call intensity, signal attenuation, the habitat structure, the receiver tuning, all of this affects masking. But we wanted to know what is the effect of chorus species composition and color density, both absolute and relative. So this is what we did, and this was here there was no field work involved. All the data was already there from the previous work, and we just put everything together in a simulation and asked what happens when you have heterospecific choruses of these 5 understory species. We created artificial choruses, so small choruses containing 20 individuals belonging to these 5 species, and artificial large choruses which had 40 individuals belonging to these species. And to summarize, the main finding of our work was, on an average, choruses that were species even, where each species had equal probability of occurring as the other one, resulted in higher levels of heterospecific masking than choruses with strong dominant structures. Now, this also was intriguing, and it was not something that I had expected. It was completely different from what my expectation was. Somehow, I thought that when you have many different kinds of colors, more diversity is better to reduce masking as opposed to having one species that dominates the chorus, but the results were quite the opposite. So the question was why? What we found by looking at our data, that the relative abundance of Mycopoda, the first species that I showed, which calls up to the ultrasonic range 80 kilohertz, the presence of Mycopoda was really the game changer. When you have species even choruses, the probability of Mycopoda occurring in the chorus is equal to the probability of any other species occurring. This results in very high masking as compared to species dominant choruses, of course, except with the exception of choruses where Mycopoda was the dominant species. So this is what we really think is going on, and species even choruses resulted in higher levels of heterospecific masking. Now, what does this mean at the community level when they aggregate together to signal? What it means is that the presence of broadband species with other naturally co-occurring species. Broadband species means those that have sound spread over a large range of frequencies. That results in species even choruses having higher acoustic overlap, and that is not hard to understand. So our predictions are that collar densities should be lower in species even choruses than in species dominant choruses. We also want to understand whether species dominant choruses are indeed more frequently observed than species even choruses. What is really happening in the natural world? And these, of course, are questions that need to be examined in future studies. Last but not the least, behavioral strategies also need to be examined if insects are exhibiting active avoidance to escape masking. Remember, we have not evoked active avoidance in any of this. Despite not evoking active avoidance in the natural choruses, as you know, we have shown that there is practically silence in a cacophony. But what would happen if these choruses were species even? Then would animals exhibit active avoidance? So these are all very interesting questions to ask as to how animals behave when they come together as a large group. Now, here is the catch. All of this that I have talked about, that there is silence in the cacophony, is when I'm looking at heterospecific masking, which means I'm looking at species A being masked by a different species. But of course, the signals of heterospecifics are likely to be different from each other. The harder problem is to examine how they avoid masking from each other when you look at conspecific masking. So this is individual 1 of species A, individual 2 of species A, individual 3 of species A. This is really the real deal. Okay, this is where, and this is very common, conspecific masking interference is possible in animals that occur in very large numbers or in groups. So bats emerging from their roost, frogs communicating in um, large numbers in a bog, or birds. And many people have examined masking avoidance in conspecific groups. So Holger Guelritz has worked extensively on understanding how bats exhibit jamming avoidance. Mark B, as we all know, has worked on frog choruses, and Thierry Aubé has worked on how birds avoid masking, or what are their solutions for conspecific masking avoidance. So the question really was, are the solution, problem is common, are there different solutions? Yes, of course, there are different solutions. Here I have listed the possible solutions for heterospecific masking, and spectral partitioning is an important way in which animals manage to reduce heterospecific masking, and by having different loudness of their calls. For conspecific masking avoidance, of course, the individuals are going to all call at the same time of the night, at the same time of the year. So the first two modalities of temporal partitioning is not possible. Fine temporal is possible. Maybe they exhibit active avoidance. Spectral partitioning is also ruled out because they have similar caller frequencies. They are going to have similar receiver tunings because they're the same species. Vertical stratification is not possible because since they are the same species, they're likely to occur in the same part of the habitat, so either the ground or the understory or canopy, but they can space apart horizontally, and maybe loudness. So these were the three things that we then examined to examine how conspecific masking avoidance may actually be happening, and it's just something that people have not examined that deeply. And this is where my PhD student, my former PhD student, Richa, examined conspecific masking interference in a field cricket. So this was done on a field cricket called Acanthogryllus asiaticus, which is found very commonly all over India. And this is what it's called, looks like. We have a lot of information on what the call call producing structure looks like, the wing of the cricket. We have information on whether the call is an indicator of bigger body size. We know that yes, the crickets which have larger harp areas have basically, we know that bigger crickets are producing lower frequency calls. We also know that the ambient temperature has an important effect on the calls of these crickets. And we know that their activity patterns is such that most of them actually call during the summer. We know that at different ambient temperature, various call parameters, as you can see, change for the species. Okay, so they are, of course, dependent on the external temperature. So, of course, it is affecting their calling features as well. We also know that their developmental temperature affects their call features. All of this is already known. We have done all of this in lab and field-based studies. So we know a lot about the species. What we did not know, of course, was how does noise affect their call structure? We know temperature affects, we know that the body structure affects all of this. But what about ambient noise from multiple calling individuals in dense choruses? So this is what we started to look at. And the first question was, is there even potential for masking? Are there crickets spaced apart in their natural habitat? If they are spaced apart, then how far can they be heard? Are they spaced? So how are they spaced? Are they calling from dense choruses, or they're calling from choruses where each one is far away from each other? So we mapped the choruses of Acanthocles asiaticus, where each individual was calling from, and we measured inter-individual distances. Then we mapped 10 natural choruses in the habitat of the animal. Each one is in the ID of a single male. So you can see there are some choruses which are very dense, there are some which are not as dense. So we mapped all of that, and we know that the nearest neighbor distance was about 3.5 meters. We examined signal transmission, and we found that the call actually transmits easily up to 3 meters before going down to the ambient noise level. And this is just to show how these choruses, the circles actually indicate how far the call of an individual can transmit. So wherever the circles overlap, this individual is being masked by another neighbor. Okay, so this is how you can now map the actual choruses, and you can see in some choruses masking is a serious problem, in some other choruses not so much. What do we find? We find that given that an average nearest neighbor distance is 3.5 meters, and that their sound transmits up to 3 meters, the active space overlap is 41%, which is, a very significant value. So 41% of their active space is actually getting masked by a neighbor. So this is a serious problem. On an average, the number of maskers that an individual has was 2. However, given how the sound transmits, the average number of audible maskers is only 1. So which means that an individual on an average can hear at least one neighbor calling and has the potential to actively avoid masking, avoid getting masked by that individual, by its neighbor. So yes, there is masking. Yes, there is a problem. And we ask now, what do they do about it? So if two signalers signal exactly at the same time, then we say there is synchrony. So there is complete overlap of signals um, in time. And if the signals fall in the silent interval between the chirps of the other male, then this is known as alternation. So we did both field observations and we conducted lab-based experiments to ask whether they synchronize or do they alternate. To what degree do these calls actually overlap with each other in natural choruses and in simulated um, masking scenarios. So this is just to show you how we calculate the probability of masking, if you like. So whether they are actually alternating. If the signals fall, this is just the phase angle between a focal animal and its masker. So if the signals fall, if they alternate, then the phase angle will be between 180 to 270 degrees, and anything on the other side will be synchrony. Okay, so this we used, and we measured phase angles in the field, and what do we find? So the median vector is around 184 degrees, which means most of the individuals are actually alternating with each other. Okay, so if you find two crickets calling together who are nearest neighbors, when you measure the phase angles of their call, so they are like calling. So it's like they are not calling together, they're not calling like this. Okay, so they're alternating with each other. This is in the field. We then simulated, so they are all alternating, yeah. In lab, we took crickets and we simulated a situation where there is a masker and we asked what do they do once they are singing solo. They are singing at a certain pace. Now you simulate as if a nearby animal has shown up and is starting to sing. What does the focal male do? What we found was it resets its call to start alternating. Okay, so it is still between 180 to 270. This means most of them alternate. So both in lab based controlled lab-based experiments and field observations, we find that our crickets are Acanthogloss asiaticus is actively avoiding nearby neighbors by alternating with the neighbor. So what we found was, yes, there is some degree of spatial partitioning because the crickets are actually spaced far apart, that there are, even though there are multiple individuals, sometimes above 20 individuals in one chorus, they are spaced far apart, that on an average, a cricket has only 2 potential maskers, of which only one is audible. We also have evidence for fine temporal masking, that the crickets are actually actively avoiding synchronizing or masking each other. So they are escaping masking at the behavioral level. And we also check for whether they are modulating. Do they show what birds show, the Lombard effect? Do they become louder when you have a loud masker next to you? And what we found was we didn't have any evidence for the Lombard effect in the crickets. So to summarize, a combination of natural spacing, signal transmission in the natural habitat, the behavioral strategies of alternating together help Acanthogloss asiaticus escape masking from conspecifics, which is the hardest possible scenario of masking that you can get. So solutions, what we find, are different for both heterospecific and conspecific masking, and it highlights the role of active avoidance, signal structure, and the role of spacing in reducing masking overlap. So we have common problem of masking, but the solutions are different. With that, I would like to thank all of you for your attention, my lab members and the funding agencies, my institute, ISA Mohali, and everybody who participated in these experiments. Thank you very much.
Thank you, Manjari, for a very, very interesting and wonderful talk. So I would like, if you have a question, please just type a question mark in the chat function and I will call you up. And when I do so, please just introduce yourself as you are continuing to build a community. So I will start with Eduardo.
Thank you, Linda. Thank you, Manjari. Very, very interesting talk. I apologize. I needed to step out. I explained to some of you that I have builders at home. I am wondering, is anybody exploring the genetics underlying both the sexual dimorphism in the wing structure and or in the behavioral patterns? And has anybody examined the development? When do the sexual differences begin to appear in the development of the cricket? And I know this may not be something you do, but I'm just curious a little bit more on the natural history and underlying genetics.
So for this species, we haven't looked at the genetics, and primarily because the wing structure variation is not as so. For instance, Nathan Bailey and Marlene Zook, as you know, the Hawaiian crickets, where they have lost the sound producing structures. There are silent morphs. So that is a fantastic model system to actually examine some of these things. We don't find these kind of things in our field cricket. So we're not looked at that. But yes, we have examined, so we know at what instar these sexual differences appear, but that is true for most crickets. So at a certain point of their nymphal stage, you can see that the wing stubs appear, and at some stage, at a later stage, you can see that these sound producing structures become evident in the males and the females also have the ovipositor. But what we are also interested in is, and what we have examined is how the developmental condition affects their sound. And we know that, and this is in the context of climate change, if you think, if you are developing in warmer temperatures, we know that, some species really, their populations crash. So they cannot even, the hatching fails, okay? And this is also true for our species. But we also wanted to know that if you do not push them to the absolute limits, So it's not like, it's 5 degrees or 10 degrees difference of their developmental temperature, but in the natural range, when you have very warm temperatures versus the average temperature, what will happen to the crickets? We know that their developmental time changes. We know that their growth rate changes. We know that their size changes. But most interestingly for me, we now know that the developmental temperature, not just the ambient temperature in which the adults are calling, so the temperature in which the young ones are growing, is going to change the song of the cricket in adulthood. So crickets that have been reared in warmer temperatures sing songs which are different from crickets that who have developed in cooler temperatures. The question now is, what about the females? Is there going to be a mismatch between males and females who have developed in different uh, environmental conditions? So yeah, we know all of this, and this has implications on how what is going to happen when insects, they're disappearing everywhere. Uh, but are they disappearing because they're just not hatching and they're dying, or is it because reproductive uh, behavior is kind of, collapsing? So yeah, but not the genetics.
Thank you very much.
Thank you. Thank you. Thanks, Eduardo. So next we have Raghavendra.
Thank you. I am Raghavendra Gadadkar from the Indian Institute of Science, where Manjari did her excellent initial work for her PhD. Manjari, I enjoyed your talk, and every time I listen to you, I enjoy it more and more. So it's really wonderful piece of work. I have a couple of related questions. You didn't say, or I didn't understand, what is the effect of masking? What is the problem with the masking from the female's point of view? And if there is some decrease in its ability to perceive, is that decrease continuous? Is it linear? If you measure the masking quantitatively, is the detrimental effect of masking linear, non-linear? Is it a step function? What exactly is the problem? And related to that is it not possible that there is inter-female variability in the ability to deal with masking? I know some people who can manage much better in parties than I can. So is that something that we need to take on? And a related question is, we're always assuming that masking is a bad thing. But maybe some males want to mask the other guy because I know I'm so much better that I would deliberately want to mask that guy and sing while he's singing, and I'm confident that I will be picked up.
Thank you, sir, for these 3 questions. So for the first one, does it affect the female's ability to locate the males? Is not something that I have tested so far, because the right way to do that would be, so if you have, we have done behavioral experiments where you have multiple sources of sound. So you have multiple males calling and we release the female will exhibit choice and will go to one speaker. We do not know that is it because she can only hear this one, which is the nearest one, or is it because of something else, right? So that's it's confounded. So unless and until we do some electrophysiology, I think that, unless we can map the neural thing, I don't think that question can be easily answered. So it's a tricky, very difficult question to answer, in my opinion, but a very interesting one.
But can you not check whether if there are 2 males who are masking each other, is the female more likely to choose one than the other? So is it random or is it non-random?
We know that they will always go for the louder male, but that simply means that the caller is being able to rise above the noise. And also, it's a double whammy for the other male, because not only is it louder for animals, it is also going to have a larger catchment area. So more females are likely to hear it. So that's why you were interested in the Lombard effect, that when you have multiple maskers around you, do the males become louder? Do they sing louder? The crickets don't seem to do that. We also know, just since we are at the topic of masking, we know for females that definitely anthropogenic noise, masking by anthropogenic noise, is terrible, because they just absolutely cannot find. They are disoriented. They just cannot find the males. In this species, we have tested those things, and we know that the behavioral hearing threshold, which on an average under silent conditions is around 40 decibels, where it's not too noisy, shoots up to 60 decibels, 64 decibels actually, which means that unless the sound is at least above 64 decibels, the female will not even hear it. So we know that masking does affect, but we know this answer for anthropogenic noise. How bad is it for conspecific noise? That is a question to which I don't have an answer right now, but it's a very interesting question. Did I address all of? Oh, yeah. And you had another question. Is it always a bad thing? No, not at all. That's the whole point. Because it may be bad for one male who is the softer male and has low, but it is totally okay for. So it is interesting to look at population level variation in the signals. How large is this variation and whether you have such variance in reproductive fitness of males. So that is something that has to be done at the population level. I don't have the data for that, but yeah, thanks. That's a great idea.
Yeah, thank you.
Okay. Thank you, Rak. So next we have Anna Marie.
Yeah, hi. Thank you for an amazing talk. I'm Anna Marie. I'm a postdoc at the Universidad Católica in Chile. I had a question also about what the function is of the calling. Is it only for finding a mate? But then when there is just so much other noise around, is it also kind of an deterrent for predators, so that the predators might have difficulty kind of figuring out one of these individuals?
Thank you. Yes, the function of the call is for mate attraction. So it is always, it is known, and we also have done those experiments. We know that the males sing, well, they sing actually 5 different kinds of calls, and it's not just one kind of call. The males sing a loud, conspicuous call to attract females over long distance. Then once the female approaches, they actively switch to a softer serenade, which is what we call as the courtship call. Once the male has successively mated with the female, it then switches to a third kind of call, which we call as post-copulatory call. We think it is in the context of mate defense. They also produce calls when males interact with each other under male-male competition. So when you put males together, but that call is completely different. That is the aggressive call, and it is completely different. When they win a fight, when 2 males fight, and one of them wins, then they switch from the aggressive call to what we call as a victory call. It's a very loud, conspicuous call, which the male produces, and it kind of, turns around, moves around everywhere to sort of display that it has won a fight. So this particular call that I have been talking about, this is the long distance mating call, and the function is clearly to attract females over longer distance. Now, whether masking has an inadvertent advantage of, predators finding it harder to find them. So this definitely has been examined in animals that synchronize. So many insects, especially katydids, they call in choruses where they synchronize with each other, and even frogs do that. And it is known that this can disorient and make it harder for the predator to find them. But in case of Mycopoda, for example, we know that it is driven simply by the precedence effect. The male that calls first is the one who's likely to be heard by the female simply because the female's neurons stop to fire after, so the polarization, repolarization. So that we know already, yeah. So yes, that is potentially possible.
Thank you so much.
Thanks, Anna Marie. So I'll take an opportunity to ask one question. Because you mentioned that you worked in the tropical region with the specific species that are there. Do you know of other studies that are done in different habitats, and do they also find similar results like you do, or is it just habitat specific that they mask only because they're in the tropics, and maybe those that occur in more open areas do something different?
So the tropics is a good place to test this simply because of the diversity of animals that you're going to find. So it is like a corner case scenario, like the worst case scenario, where you have so many species and so many individuals of each of the species which are packed together. But it has been acoustic masking interference has been examined in temperate regions as well, and not only in insect but also in birds. And definitely, we know that the problem of masking exists even in temperate regions, but the degree to which the problem exists is likely to be the severest in the tropics.
Okay, well, thank you. So we have another question from Rag.
Manjari, I have a question and a suggestion based on your reply to Anna Mary's questions. The question is, what is the function of the victory call? And the suggestion is that one should look at the effect of the presence or absence of the victory call in the development of winner-loser effects in the cricket fighting. So I'm postulating a function.
Yes. So we have examined two things in the context of male-male aggression. We have examined the winner-loser effect in the species, and we know that the winner effect does not exist, but there is a loser effect. That is one. Second, we also know that age is the strongest predictor of fight outcomes. It is not the size, it is not the body mass, and it is basically older males who are more likely to win fights than younger males. Now, are older males more likely to produce victory call? This is confounded by the fact that they are the ones who are likely to win. So we are going to find victory calls being produced mostly by males who are older. And so therefore, yes, there is going to be differences, inter-individual differences in the likelihood of them producing victory calls simply because, it depends on who wins the fight, and older males are more likely to win the fights. But what exactly is the function of the victory call is, in my opinion, a non-trivial thing to establish. I can hypothesize that this is in the context of mate defense, but males also produce this post-mating call that I talked about, and also this victory call, which structurally they are actually quite different. But I suspect that it is a display call basically for the in the context of male-male competition to keep other males away. And also post copulation, they really guard the female. So there's a lot of mate guarding. They continue to produce the post copulatory call, and they stay around the female. The victory call, however, is very short lived. They win a fight, they exhibit the victory call, and the show ends there.
They can't help it.
Yeah,
But if it does help in creating the winner-loser effect, then it would be a very specific function, and perhaps this should be investigated across species. So there may be some species in which there are strong winner-loser effects. There may be others in which there aren't, and then that may be correlated or may not be correlated with the probability of giving victory.
Again, another very interesting idea. I should have kept a notepad and a pen next to me. Thank you. Yes.
Fortunately, we're streaming on YouTube, so you can go back and listen and get all of his suggestions from there. So I don't see any questions on YouTube. Does anyone else have a question or comment? Or Eduardo?
Well, only because since nobody had questions, I figure, as we go into the session of questions and answers, sometimes we allow ourselves in fine to really push the boundaries and force ourselves to think outside what we've been doing. So my question is, and maybe I missed it, I apologize that I need to walk out, but the way you have shared such fascinating, but by the way, playing back the sounds in the forest, or what you alerted us that it was very loud. It's fascinating. I feel like, all of my natural history background comes to life. I love it. Now, if I followed your thinking and the way you've structured your research program, you start from the assumption that there is interspecific competition of sorts around the acoustics. And what I'm saying, and that's what I'm saying, this is not the question that I wanted to ask first, but and if you were forced to imagine a world where there's a third or a fourth factor that's responsible for causing this that has nothing to do with interspecific competition, could that be, again, I mean, I just wanted to, I'm thinking of my own limitations every time we try to explore aspects of the monkey biology. We're so dogmatically prepared to assume that temperature and moonlight are factors. We start from there, and the moment we start from there, that defines the scope of exploring, right? Because then we're leaving everything else outside. We're committed without realizing to this. Can you completely speculate, entertain, if you were forced, you're offered a $1 million award if you come up with hypotheses that have nothing to do with interspecific competition? What would that be?
So the signaling activity per se, whether it is affected, say, for instance, by predator abundance or by moon phase, I can tell you that the moon phase has nothing to do with the signaling activity. We know that. We have measured it, and it is not strongly correlated with moon phase. On the other hand, during full moons, predators are more likely to catch them as compared to new moon, but that is also because we can see that better. So these are natural history observations. So it's confounded by our own, limitations. So I would not draw my conclusions from there. As for what else could have driven, so this is all about signal structure evolution of their signal structure. What could have driven? So sexual selection drives variation in signaling traits, but at the community level, them signaling together, why are they calling with this specific group of species? Why not with another group of species? What would happen if the choruses had a different composition? This is what we addressed there. And what we now know is that species, even choruses, what I had thought would be good, is actually not good for communication. What we don't know is large scale data is what we need to see natural choruses in this kind of habitat. Are they actually species even? Or are they species dominant? So this I can tell you, I'm very happy to tell you that while I moved on to other things, my PhD advisor has now a student who was my master's student who has now joined her for his PhD, is now examining these. So to see for communities which are dominant, having dominant individuals, so your species dominant choruses versus species even choruses, what is really going on there? Maybe in another 2 years, we'll know the answer to that question, but it cannot be seen in exclusion with other factors. Of course, there are opposing selective forces that shape any trait. So certainly there are other things. But if you ask me that are species that are taken from completely different kinds of habitat who do not co-occur, are they going to have more similar structures? I think yes. So I think interspecific competition has to be, and it seems to be, one major driver of this over dispersion of the signal structure. So the strongest possible explanation for this signal structure that we see has to be competition for acoustic space. I know that the signal structure is not driven by acoustic adaptation. It is not driven by habitat structure. It is not because they're optimizing transmission. In fact, the space from where they call oftentimes is the worst possible space to signal. There are many crickets that call from the ground, and ground is the absolute worst place to call because your sound is not going to transmit that far. So why not call? If you just climb up, and I have made these measurements, I know that if they climb up just 1 meter, then they can double their transmission distance, but they don't. So I would still, not romanticizing my results, but I really strongly feel that it is the interspecific competition which has shaped the signals in this community.
Thank you.
Thank you. We have a question from Maren.
Hello, my name is Maren Hook. I'm from the University of Derby. I have normally nothing to do with cricket, but we run a cricket practical in one of our behavior modules as a leftover from my former colleague Karim Wahed. And what we and this is done on Gryllus bimaculatus, which is Mediterranean species. What we find is when the males are chirping along, so we do mate choice experiments, and sometimes it's because you said that the females always choose the bigger males. Is there also the male choosing females? Because what we sometimes found was that sometimes a male could sing along as much as he would, and the female wouldn't be interested. And sometimes it was the other way around, that the female showed that she was interested, and the male just didn't do anything. And then we put in a second male, and the second male didn't do anything, and the third male. So and these were all virgin crickets of the same age. So is there something other way around going on as well?
We know, I don't know about which species of cricket you're looking at, but in tree crickets, for instance, that produce nuptial gifts from the maternal gland, they produce a substance which females feed on. We know that the nutrition environment can even result in what is known as sexual role reversal. So, in fact, males can become choosier and may not. Also, if the males are, I don't know what kind of setup so you are doing these in boxes?
It's a student experiment, so it's a very, very simple setup, and yeah. So essentially there's a little tiny plastic box, and there is a male cricket and a female cricket originally, until the first cricket mates with a female. And then the students have to measure how long did it take for them to mate. And then we either put in simply a second rival male to see what happens then, or we take out, and how quickly does the second male mate with a female if the original male is still in, or we take out the first male so that there is only the new male, and then how long does it take him to mate with a female. So essentially it's about the interaction between the males and the female.
I'm just thinking, another reason why the males may not be, they're not even singing, I'm guessing. So this can happen, at least in our species, the males do not exhibit calling in the first 2 weeks, or at least it is really reduced in the first week. And it's only from 2 to 4 weeks when they show their maximum reproductive behavior. So from species to species, this may vary a bit. So immediately after molting, they do not, at closing, they do not sing. But after 1 week or about 8-9 days, they start singing much more. So if they are really young males or if they are really old males, that will also matter. So although all of the males were of the same age, I'm wondering if they were all very young or maybe too old.
I mean, I'm less interested actually in our cricket, but more interested in whether something like that happens as well in your crickets, that there is choice of the males for certain females rather than the other way around.
We have tested males only when they are virgin males and when they are more than 2 weeks old. So they are motivated. And what we find is that we have looked at calling activity patterns. So yes, there is a variation. There are some males who call much more than the others. We know that infection level affects their calling activity. Calling activity really crashes when they have bacterial infection. But we have never found that motivated males who are otherwise singing, when you place a female, that they will not exhibit courtship. They will usually, after antination, immediately switch to the courtship song. And I would expect that in most, unless they are providing some sort of investment, I'm guessing, which is limited.
No, they don't have any nuptial gifts or anything like that. Thank you.
Thanks, Baron. Do we have any other questions or just a general comment? Okay, so with that, I would just like to thank Manjari again for giving us such a wonderful talk and for taking the time. I know it's very late in India right now, so thank you so much. And I think we all learned something new today. So just a reminder, as we mentioned earlier, the next FINE seminar will start in February, but for now we will release the program sometime early next year. So I'd just like to wish everyone a very happy holidays, to spend time with your family, and we'll see you in the next season.
Very
Thank you.
quickly, to those of you who are regular attendees, which I think it's the case for most of you, we're already taking in suggestions of possible speakers for fall 25. So a year from now, the spring is pretty much full. But please don't hesitate to shoot me a message. If you have any suggestions, I'll bring them to the table for the organizers to consider them. I mean, I mentioned it to you because all of you, Linde, Maren, Rag, and Marie, you're regular attendees. So thank you so much. And
Would
Marie,
you be interested in machine learning?
Sorry?
You remember Charlotte Wiltshire? She's nearly finishing her PhD, and she has done some very interesting things on machine learning to identify gesture in chimpanzees, and she's very good at talking. She
Send me a
just gave
link.
it
Send me a link to her name or something like that. And Marie, are those fig trees?
It is a fig tree, yes.
Where are you?
I'm just on my balcony of my apartment.
Okay. Do they fruit?
Yeah, we have around 8 on there. They're still green though.
Good, good. Well, thank you so much, Linde. Congratulations again on your PhD. And thanks for joining us. And we'll see you back in February.
Mhm.
Wow.